This is a production of Cornell University.
So, well, thank you for this invitation. I have to give a bit of a disclaimer that so I'm going to give really it's kind of a public relations event. So I'm going to give a talk that is a version of a talk that I gave at the BTI. I'm very well amplified, it feels. That it's Magdalene Lindberg who invited me to come and give it again here. And so I'm going to point to one vision of the opportunities and try to illustrate it with projects that I know of around campus, around heavily the College of Engineering, but also physical sciences. And I'll put in a few projects that from published work from other places in the world. But I'll start out with a naive outsider's perspective on why plant scientists might look more to these emerging and future physical science tools. If I'm offensive, it'd be good if you told me, because so I won't continue to be offensive about my assumptions about the state of plant science and important questions. So I come to this having interacted with plant scientists here and elsewhere, but also having been part of a big growth area at Cornell in looking at engineering questions in biology. And I was very naive early on in thinking that, but not completely wrong, in thinking that there are a lot of similarities between the organism scale as well as the cellular and molecular scale of mammalian systems and plant systems. Now that's of course a very coarse view, but in terms of technology development, there's reason to believe that the analogies can be powerful, and there may be translation from one to the other. Now, why bring up mammals in a plant science department? Well, it's because the mammalian biologists, in particular in the biomedical field, have heavily engaged engineers in technology development to a level that I think is, well, maybe scales with the size of human biology relative to plant science, but there's been a lot of technology development in these past decades. And so one way to say is, why not bring nanotechnology and other technologies to plant science? Well, one is that they've gone to human biology. They've gone from being molecular tools through cellular tools through being clinical tools that are interfaces with the living organism, so it doesn't have to be dissected. And I'll point to some of these examples that ought to be able to be translated in some form to plant biology and agriculture. Now, working in attempts to bring technology to plants, I understand also that there are important differences that in some ways it's harder. Plants don't let you in as easily. They don't have soft skin often. And disrupting them can have dire consequences that, and in some ways mammalian systems are more agreeable to some of these interventions. Okay. I highlight the hypodermic needle because this is one of the absolute workhorses of biomedical intervention. And I would argue that essentially we don't have a hypodermic needle for plants. And there are important reasons why we don't, but it'd be awful nice if we did. So agriculture, I think a lot of people think of engineering in plant science as being things you stick to a tractor. And I think that's been the definition of for many decades. And it still is an important target. And one way to see it is one thing that has surprised me. I was first surprised to know how heavily agriculture has invested in smart irrigation strategies. 10 million hectares is a reasonably recent estimate for globally. And yet almost never is that on a closed loop feedback, because they don't ask the plants how much water they want, or ask even the soil And so here you have what is an advanced approach running free, running on somebody's intuition about what should be going on in terms of irrigation. For example, and these are topics that I'm interested in, it's uninformed by parameters that are relatively coarse but have been shown to be crucially important, like water potential, stem water potential. That it's been shown that crop productivity, quality correlate with this one simple seeming parameter in the plant, and yet we have no practical way to go and ask even a sampling of plants in the field what their water stress levels are. Today, the techniques are now mostly 60 years old and coarse and manual and hard to use, not giving you real-time data. I think there's important advances have been going on in remote sensing. I think that, and they can give beautiful information. I think what I'd like to see is bringing into the plant, getting local physiological data into that potential feedback loop. Another place that I've been learning more recently what the realities today are about breeding programs and selection of better crops. And it seems to me, a beautiful challenge for the engineering community that most engineers have no idea about. And it took me a number of years working with plant scientists before truly learning the realities of what it is to select a better plant. And so here I'm showing science fiction, a patch on that you could, a few cent patch that you could go along and put on many crosses, for example, and get something local, physiological back during a growing season outside. And it was actually partly meeting with Ed Buckler that crystallized this challenge for me. And this is a paraphrase, but he basically told me, if you can measure a trait 10,000 times, I'll find you the gene. And you know better than I whether he's correct about that. It looks from his publication record like it could well be true. And again, this challenge, thousands of reproducible quantitative measurements on plants, that's a great challenge. And it's one that engineers should know about, should be going after. Because I think it's possible, not for every definition of a trait, every phenotype, every protein, but for some crucial ones. And this, I gotta say, I saw a talk by Pioneer, it was maybe he was retired from Pioneer, and he admitted that, in his opinion, leaf angle had been the biggest change in phenotype that had defined increases in yields in their breeding program. And that may be an oversimplification, but I was shocked that that's what that these extraordinarily sophisticated molecular biologists deal with extremely coarse physiological information, extremely coarse definitions of what a trait is. And I can understand why, because you have to measure it thousands of times, you have to measure it in complicated environments. But again, it's in some sense, it's the engineer end of the problem that's failed in this project. Because I know that the molecular tools are incredible, but if you're applying molecular tools, the absolute state-of-the-art in genetic analysis to leaf angle, that seems unfortunate. So I know. I have a very naive, again, perspective on what the molecular and cellular questions in plant biology are. And so I'm just giving one example here where I see opportunity. And again, I was surprised to learn about the state of knowledge in plants. Here's an example of it's a review saying that direct evidence of the movement of a peptide has yet not been achieved in the lab, and that this particular flowering locus T in 2013 was said to be the only example of long-range signaling by a protein in a plant. And this seems like, again, this is a challenge across biological fields, but to integrate information, extremely sophisticated information about individual cells and molecular processes with organism scale processes. This is a place where in biomedical engineering, it's been a rich area for making progress to get to the tissue scale, get to the organism scale with hypotheses with molecular hypotheses. And so it's another opportunity. So I'm going to talk about three perspectives on how, and I'm going to illustrate them with lots of people's work. I'm going to talk about what I call devices, and this is where Cornell has been a leader in developing small-scale devices, and a lot of that energy in the time I've been at Cornell has been pointed towards human health. Not much of it pointed towards plant science, and I think that's a missed opportunity to date. Cornell is also extremely good and in imaging, and I know many of you probably use advanced imaging techniques, but I think this has been continued to be leaders in the world in advanced imaging techniques, and then also models. There's models that I'm going to focus on, not bioinformatic-like models, but more what we call systems biology models, which again look to integrate information from the molecular across to the cellular and to the full animal, full organism physiology. So I'm going to start with devices, and I'll just point out that Cornell has a tradition in this. One of the workhorses of soil science and agriculture still today was invented here. There's some argument about precedent, but the Richard soil tensiometer is still the only tool to measure precisely across the plant physiological range of drought stress. The few bars of tension that are relevant to most agriculturally relevant plants, this is it. Versions of this technology. I think most of you know the gene gun, and of course, both of these came from cows. They didn't come from the engineering college. So that's, I'll admit that. But I think these are two technologies, historic technologies that came out of Cornell that are precedent for what should be being done here still today. So I'm going to turn to a number of technologies that I don't know of being used in plants, but I think could be interesting. And so this Erwin de Valmek is a new faculty member in biomedical engineering, and this is something he's working on today, and this is a publication he published just before he came here. And this is an example of using a microfluidic system, which is a miniaturized lab that can handle extremely small-scale samples to collect single-stranded DNA out of the blood. And it turns out that has an important in the context of transplant biology of transplants. But this is one of many examples of doing molecular biological analysis on what would have previously been thought of as being unusable samples, the contents of a single cell. You can do analysis on the contents of a single cell today. That's still a challenge, but it's the kind of advance that is represented by nanobiotechnology. This is an example from Harold Craighead's group in applied physics. He is, I think, maybe the person who coined the name nanobiotechnology when he started the Nanobiotechnology Center here at Cornell. And one of the big themes has been manipulation of biomacromolecules, and in particular DNA, that has led to the beginnings of, what is now becoming a reality of single molecule sequencing. And this is an example, I believe this is an example of mapping epigenetic change at the single molecular scale. And so what you see here is this is a truly, here are the features that were created in Cornell's nanofab are of a molecular dimension and forcing this DNA to take on a conformation that allows it to be literally read with microscopy. This is another example from applied physics of creating a cell culture that allows electrophysiology to be done with subcellular resolution. So this involves both the traditional patch clamp type approach, but also the cell that is living on a circuit that allows the group to interface with it and watch for events that occur to triangulate to where these events are occurring on a subcellular scale. Moving up a little bit, and a number of these ideas are about cultured tissues and cells, and I know there's a difference in how important that is in plant biology than in mammalian biology, but I think I hope that some of these examples trigger some ideas about where the translation could occur. So this is an example from Mike Schuler's group, who I still claim him for my department. He's actually shared with biomedical engineering. And this is an example of, in fact, building a mimic, a replica of the connectivity of tissues in a mammalian system. And so you see here liver, fat, kidney, and coupling them with a very primitive vascular system, but one that allows the metabolic environment and the signaling to interact with these different tissues in a manner that simulates timescales and transformations that occur in the different tissues in the body. And so this type of replication of tissue properties, of the tissue environment, is something that there's a lot of know-how now, both from the material side, the fabrication side, and in some cases the biological side, about what are the influences of the tissue environment and the communication between tissues within a full organism. This is an example, again, that is called a tissue engineering effort from my lab of a focus on the microvascular structure of humans, which is, like the phloem in a plant, is built of living cells. Okay? The vessels on the smallest scale are really formed of a single layer of endothelial cells that have a huge amount of biology associated with them. And of course, it's a bottleneck for forming any big tissue is to simultaneously form its vascular system if you're going to do an engineering process to form a tissue. I generally, and I've spoken to very few people about this, but I wonder, it would seem naively that tissue engineering might be a whole lot more fun and easy in plants in some ways. That creating, and because plants might just go with it. And there's not the clinical vision of bringing somebody a lab-grown heart, but actually today much of tissue engineering is done to recreate a multicellular structure to study its biology at that multicellular scale. There are very few examples today of translation truly to the clinic. And so in that same spirit, I could imagine that it could be interesting for in plant biology. Here's an effort that I thought was exciting from Northeastern, not from Cornell, but it's another example of creating a microenvironment. And in this case, so again, what we call a microfluidic system that has microscale features that, inside this cartridge. And this is, by creating that environment and burying it in the soil, they were able to grow up a huge number of uncultivatable microbes. And from that, they managed to isolate promising antibiotics that had been unseen previously, because if you just took the soil, you had too little of any one species to produce that small molecule. And so this is, I wish it had been an idea here, but it's an example that I think it's worth noting that one place, and in fact Harvey Hoke, as a plant pathologist, was one of the early people that interact with the Nano-biotechnology Center, one of the most active biologists at the Nano-biotechnology Center. And one reason to recreate these mimics or replicas, these engineered tissues, it may be more about pathology than it is about the plant biology on its own. Okay? Here is one of the first efforts, first targets that my group went after was to build synthetic xylem. And because plants do something that engineers don't, that humans basically don't, which is use liquids under tension. And so we made very, very simple replicas, and it was really more about a materials question of how to build a network that coupled to the outside in a way that allow the fluid in these tiny vessels to go under tension in the manner that's proposed by CT, cohesion tension theory. And it turns out that we in a synthetic system validated cohesion tension theory, which has nice consequences physically. It brings us into a new regime physically. But I think it also gives us an opportunity to, for example, understand the design ideas from the purely physical point of view of here, for example, the segmented xylem structure. Seeing these events of embolization occurring independently, as has long been proposed for the redundant segmented structure of xylem. I've also thought that, and I've spoke to Harvey about this before he retired, that this creates an unusual environment, the environment of the xylem in the leaf, for example, where important pathogens live, that is unlike any other environment in the world. So what is it like to be a microbe that lives with the fluid pressure around it going from 0 to minus 20 bars up and down and up and down. And so it's also potentially a bioreactor environment to study the biology of plant pathogen interactions. One of the things we've done with this, and this was Alan Laxo, we were building a device that extrapolated from our synthetic tree to have a cavity of liquid and it's a strange geometry, but this gray area around is a nanoporous membrane that allows the liquid in this cavity to go to tension when it's exposed to a dry environment. And here we were building it to have direct mechanical sensing so that we'd have a direct measure of that stress. And Alan came to find out what this synthetic tree was and so on. And when we showed him this that we were building as a laboratory to study liquids under tension, he said, oh, well, that's a tensiometer. And we hadn't known about Richard and the whole challenge of tensiometry till that point. And we realized that this was, in fact, a miniature Richard's tensiometer. And again, like I told you, we've come to appreciate that tensiometry, measuring drought stress, as a tension, is the most sensitive way to measure that thermodynamic state of water. And so having here a strain gauge on a diaphragm over this cavity allows us to have quantitative measures of the water potential in the environment around this device. And in this case, this is our champion of this generation of these devices. We measured, it went to an embolization event at 300 bars of tension, or water potential of minus 300 bars. And just this is, I don't know if this will, yeah, maybe, to actually see, I don't know which of these work, but to actually see embolization is fun. That this is the release, this is the click that acoustic measurements here, this dramatic release of the tension upon embolization. And so it turns out we've been continued with Alan and Taryn to pursue this so that it's truly practical for plant science measurements. And so here is our current generation of this device. which is, as you can see here, down to a relatively small package. I'm failing to get a pointer here. This in this format, it's like a pinky fingernail. And it turns out one of the nice things is that I like is that to make it respond fast enough to be able to catch all of the changes in water potential that might be occurring in a plant, which is where we'd like to take Richard tensiometer and move it into the stem instead of being purely for the soil, we had to give it a vascular system. We had to connect this cavity to the outside world with vessels that would, as in plants, as in the xylem tissue, transmit the stress, maintain the stress. So they're divided from the outside world by membranes, by nanoporous membranes. But once inside this device, fluid moves extremely rapidly along these vessels. And so the idea of segmented xylem allowed us to take from our first prototypes, our first generation of these devices, very long transients that would have meant we wouldn't capture things that occurred in less than an hour, to having just tens of second transients that allow us, we think, to capture all diurnal and so on behavior of a plant, and to do it across, bring the Richard tensiometry idea out towards 100 bars. So across all the way to really desert stresses in plants. And we actually have just moved this into plant tissue, and we have trees growing in the greenhouse so here, working in branch segments, these are actually just in the last week to actually measure. Here we apply drying stresses onto this branch segment and can watch what we've been calling in the lab diurnals, although they're not, they're just tens of minutes long, and they're associated with not with true transpiration, but with imposed transpiration. But we are hoping that this will allow us, in an embedding process here designed for a woody species that just takes a few minutes to get real-time, long-term stress information on plants, and as part of water relations research, and eventually as part of irrigation scheduling, to close that feedback loop with what is a very relevant way to do it, the plant's actual stress. And this is just a zoom to see we're able to resolve on tens of bar scale. We can resolve small fractions of 1 bar changes in water potential, which, of course, for annual species may be the more relevant scale, just a few bars instead of tens of bars. So that's an example of actually bringing one of these technologies to a plant. Here's coming to a leader in the world of making electronics, sophisticated electronics, compatible with life, John Rogers' group, which is now moved to Northwestern. And this is an example of what I think, I pointed to the idea of an RFID chip or patch on every leaf of a field of plants. And this is an example on human application of a real integrated circuitry on a flexible adhesive patch that gives you, in this case, electrophysiological data through the skin of the wearer. And so I don't know of anybody moving this into plants, but if, again, with the right inspiration from you, I'm sure it could happen. Moving towards the field, bringing these things from research questions to the field is certainly a place where there's lots of expertise on campus. You may have seen this. This is a work from the Ericsson group in mechanical engineering, where they have turned a iPhone into a relatively sophisticated sensor diagnostic system for metabolic diseases and symptoms. And so this is an example of engineering all the way towards something that you can hand off to a non-expert user and bring to the field and do diagnostics. And again, there's this type of idea once you have the idea of a lab on an iPod, or iLab on an iPhone, you can, there's no reason not to bring it to a question about a biochemical question in plants. We have a very large, very strong electrical engineering department. This is an example of Edwin Kahn, who is very much focused on this question of completely wireless, power-free sensing. So all those patches may not even need a battery. As with the checkout counter RFID, an RFID is just an antenna. It picks up the energy it needs to respond to you just from the radiation you send at it. And so this, again, is is someone who'd be, if you have sensors already, and what you need is to have 10,000 of them that all talk to you, there's that expertise on campus. So that was my longest section. I'll just give a few examples of imaging capabilities. We have one of our imaging experts here, Becky Williams. There is a very good shared facility in Weil and in biotech building. I'm going to highlight one of the things I've found very exciting recently, which is a collaboration of Matt Patrick and Warren Zipfeld. They're developing advanced versions of the microscopy that I'm showing here. And this is a mammalian example, but it's a super resolution technique. And what we're seeing here, it's hard to maybe read this scale bar, what we're seeing in the Z direction, deformations of the cell's plasma membrane of tens of nanometers. Okay? And so this cartoon, the bumps in this micrograph are associated with individual or small clusters of transmembrane binding proteins interacting with the substrate. And so this is one of the things, this is going from pure structure to dynamics in the current efforts. And it's a huge possibility to watch interactions at the plasma membrane, one molecule, one transmembrane protein at a time. This is an example that you may have heard about recently. Uli Wiesner got a very large National Institute of Health grant for this effort. It's in this case, the star of this picture is this little sphere here, which can be just a few nanometers across, and carries with it fluorescence and chemistry that allow it to be a detector. Okay, in this case, they're using it to carry contrast agent for an imaging, but they've worked on this in many different directions. These little lab on a particle is an opportunity to have a truly dispersible sensor that could go to the tissue in an intact plant, go to the tissue where you want it, and then be read out spectroscopically, for example, or by fluorescence. An example, not from Cornell, but from this field of so-called nanomaterials, here from Michael Strano's group at MIT, of disrupting a chloroplast with a nanotube. And what he finds with my simple understanding is essentially by putting this carbon nanotube, which is just a few nanometers in diameter and has tunable electrical properties, he's essentially putting an antenna into this organelle within the cell. And this is him piercing a single chloroplast and getting an infrared signal from that chloroplast that was invisible before he put it in. They go on in this paper to claim that this is improved photosynthetic efficiency in these leaves that were treated with. I have some worry about that claim. But the idea, again, of having almost molecular scale engineered material that can be part of transducing information from the cellular scale or even influencing the behavior at the cell and subcellular scale, I think is interesting. So models, again, I'm looking towards models that I know there's a lot of expertise in CALS and in plant science, in bioinformatic modeling. I'm less familiar with modeling of gene expression the kinetics of gene expression coupled to signaling, coupled to cellular behavior, coupled to transport processes on the organism scale. And that's one definition of systems biology that I think is very relevant to plants. And so Mike Schuler, again, here I gave him both his departments, biomedical engineering and chemical engineering, is actually famous in this community as a very early developer of complete treatments of cell behavior. Here, starting with microbes, bacteria. But what I mean by complete, in this case, the definition of the complete is that he has a set of differential equations that are self-sustaining, alive, and respond to metabolic stimuli and so on in a way that is reasonable, including gene regulation. Okay? And so going from gene regulation, gene expression, through to metabolism, those are tractable. There are a large number of tractable models for prokaryotes and eukaryotes to be worked with, to ask questions. Jeff Varner in my department is somebody who is one of the people who push this to extremes of completeness in terms of the number of molecular players they're willing to put into such a model of the cell. Again, going from gene expression, signaling internally and with the external environment. And in this case, moving to a multi-scale consideration of how a cell evolves in its tissue context. And so with signals coming from other parts of the tissue and through vasculature, for example, other parts of the organism. To highlight an effort with Bob Turgeon that I've been involved in on the questions of flow and loading. And here's an example of where focused on the biochemistry associated with flow and loading and transport questions, very detailed questions about plasmodesmatal, the physics of plasmodesmata, and how they regulate what gets loaded. And we do this, we claim, and I believe that understanding the implications, the design ideas that are represented by plasmodesmata requires some accounting for the full organism scale coupling. That this is what the concentrations, water potentials that define transport very locally in the at the mesophyll phloem interface in the leaf are coupled throughout the entire plant. And so that again is an example of where different levels of what could be defined as a systems biological approach can lead to hypotheses that then lead to experiments, and it's a useful iterative process that I think is not done enough. So they're here, and I can share these slides. There are a number of other people across the university that most of whom have not thought to apply their skills to plant science. But I think a number of them would be happy to. And there are, again, lots of resources. This talk was originally a co-talk with the Center for Nanoscale Physics, Nanoscale Science and Technology, the CNF. But I'd also point to and I believe they're represented here, the Center for Material Science, which is, like the CNF, an extremely robust, long-term effort, which in particular, for example, has some of the best microscopists in the world and the absolute best TEM facilities that can be had in academia. And this is actually Lena, is recently arrived and recently acquired a $3-4 million transmission electron microscope and is very interested in biological questions. So to summarize, there are opportunities that go from the very most basic molecular biology through to something that will be coupled to something strapped to an attractor. I think and it'd be very nice to see the kind of interactions, vigorous interactions that have been going on between engineers and human health biologists, going on between engineers and plant biologists. If there are any decision makers in the audience, one of the things that helped a whole lot early on were seed grants. We had no collaborations with Weill Cornell Medical School. And then there started to just sprinkle on these 50K seed grants once a year. And it was amazing how quickly that changed. Now, of course, biomedical engineering also grew a whole program. But I think there's reason for this kind of interaction, I think could lead to a lot of activity. So with that, I'd like to thank my group, a number of people, a number of collaborators, including those here. I think, Bob, I have not gotten you on my collaborator list yet. We got to get those papers out, but that wasn't because of that. But, so thank you very much for your attention, and I'll be happy to take any questions.
Well, there are two sensors in plan for redox potential, for redox potential, and also pH and other sensors that are built into plan for that's for engineering.
So it's encodable sensors.
Yeah, then we put into plan.
Well, that's great. But it's not always that easy to read them. And so I think Becky and Warren Zipfeld over in are working on ways to read those sensors, which are finicky. And of course, always the more photons you can get per sensor, the better, and working out tricks to calibrate them and so on. So that's a good theme. And I agree. If you can grow your sensor into your plant, express it in the plant, great.
Is there an impediment in the excited or flow for small particles traveling into the cell, through which don't have in animals?
I do think there are physical size exclusion steps that the animal cells that line the vessels actively endocytose components that, at least in the context of xylem, would not make it into the size, not make it into any size at all. They would be size excluded by mortar pits, for example. And challenging, they would be size excluded from entering the room. And so but what that dimension is not, I think, is somewhat species dependent and not always well characterized. But I think some of these nanomaterials are small enough to be taken up. Somebody in the audience may know better about uptake of particulate, but it is a trick, and that's again, and you can't just use a hypodermic needle to go into the xylem, or the foam for that matter. It'll stop working. So that's a frustration.
So you mentioned kind of the differing scales of plant versus mammalian trials. I mean, like a small corn breeding trial might be 10,000 plants, which would be an absolutely massive trial on rats, much less humans. I guess, what are kind of, in terms of these technologies in mammalian systems for tissue sensing, what are the largest scales that people work on, and how could that be modified for the scales that someone working in like the cereals might be working on?
Well, so there are two. So if you, given the budgets I hear for breeding programs, you could get people interested enough to make enough sensors that their cost would go very low. So if I could make enough of my xylem sensors to satisfy breeders, if they have a way to use them, I could bring the cost of that sensor down to very inexpensive because it benefits from the scaling of price that microelectronics benefits from. So that's one. If there is a real investment, you carry a supercomputer in your pocket for a few $200. So that is present. The other perspective is that some of these approaches around so-called nanomaterials, these can be synthesized by on the many gram scale. And so that they can be, if the delivery can be worked out, that and there's one reader strapped to one tractor might be able to read all the plants, and you have to paint every leaf, but you must be doing something like some such human interaction with every one of those 10,000 species right now. So it's not today. I don't. If I had it, I'd be doing something with it. But the 10,000 replicas of an experiment out in the field is a challenge, but it's a good one. Is what I'm saying. And I'm hoping, I'm trying to articulate that to this community and to the engineering community, and hopefully to the funding community, that there'll be an investment, because I don't think there's no reason it's impossible. But what does the engineer really need from the plant biologist is what to measure. And it's not just your dream of what to measure, but what is it to come to a compromise between what would be the perfect piece of information and what would be scalable to 10,000 replicas at a reasonable price. And so, and my feeling is that the breeders are quite good. They don't think about it. They don't, and this is the gene to trait gap. They're so focused on the gene identification that they're not worrying much about what traits might be good if they could get beyond leaf angle and so on.
It seems like being able to tap into the flow and sense small molecule signal through the flow would be a game changer for understanding what's happening in the plant. Certainly in plant-micro interactions, there's a lot happening there. And there are curious things that one could potentially build on. For example, some insects are able to tap into that.
That's right.
You can hijack that. But are there any ideas you have on?
Well, we've explored some of these in the CNF, for example, of trying to, so the proboscis of phloem feeding insect can go through without inducing clotting. That's the right word. And so this ought to be replicatable. And there's been a huge effort on microneedle drug delivery for human tissue. And so it's extremely fine. So it's like a pulled pipette, but replicated many times over with tunable chemistry on the surface. So yes, I think there is a possibility there. And I think looking to the biology, so I think also species like the mistletoe that grows itself into tissue without disrupting tissue, the biochemistry that allows it to do that gracefully, the suppressors of plant response, those are part of, I think, of what's going to have to be allowed for a graceful interface like that.
Well, you have another question? Yeah, okay. Another question?
Yeah. So you focused a lot on mechanical properties, but of course plants are engaged in a lot of exotic chemistry as well. Have you thought about sort of technologies that might be used to sort of make sense of discovering novel compounds or something like that?
Yeah, absolutely. And so for example, and in the examples I've shown, much of the focus has been is on biomolecules, macromolecules, and small molecules in terms of analysis with small samples on small scales and cheap devices. That is a big part of what the so-called lab-on-a-chip effort has been. And so whether it's the volatiles around the plant or small molecule signaling within tissues, it's a good target. It's not one that my own group has been focusing on, but there's a scaled down.
So I'd like to say that I think that Abe has given us a number of really excellent illustrative examples of what can be done, as well as some really stimulating ideas of opportunities where I think our community has to maybe do some more thinking about how we can address some real breakthrough new advances by bringing together our problems with, in a sense, Abe's tools. And before thanking you, don't forget that we'll have a session out in the hall where we can continue discussion with Abe and also with others from these other facilities.
Well, it'd be great to have a volunteer to come do the reciprocal talk in engineering. Or volunteer somebody you know. But I think it could be, because again, they talk to students and they've heard about the biomedical challenges, they've heard about tissue engineering, they've heard about cancer, and they know less about problems from agriculture and plant science. And so it'd be great to do the reciprocal talk, and we could find a good forum for it.
Great. Okay. Thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.